Well, thank you very much, Kristalina. That was overly kind. I actually have a picture of you later, which I'll be revealing. It's all right, there's nothing dodgy. Anyway, thank you all for coming. What a fantastic turnout. I'm absolutely delighted to be here. In fact, I was just talking to Paul, the chef, who knows that I come here at least once or maybe twice a term, so I'm getting in big trouble with other colleges for turning up way too often to Reuben, because it's such a wonderful place to come and to host dinners. Anyway, I'm delighted to be here tonight. I'm going to indulge myself and mostly talk about my life as a scientist and the work that I did over the best part of 35 years in the neuroscience space, trying to understand this really complex, both everyday human experience and animal experience of pain, and then also it as a very significant major medical problem, a little bit on consciousness. But then I'll give you, if time permits, a bit of the backstory, because of course, alongside delivering the science and the excitement and the love and the passion and the curiosity we have as academics, whether you're in whichever discipline you are in, there's an awful lot of stuff that goes on in the back, your personal life, as well as just building things in order to do the science in the first place. So I thought I would just share then a little bit about the journey and just a few key thoughts that have been really important to me in the best part of 35 years. And of course, 3 years in now as Vice-Chancellor. I'm not going to talk about being a Vice-Chancellor. I'm not going to sort of wow you with how fabulous the university is, blah, blah, blah. We can get into any questions you want in terms of my role and that hat that I wear over Q&A. And please know that you can ask me any question. There's no questions off limits. But mostly this is going to be about pain and my life and the pleasurable sides of my life. So I was admitted on the January the 10th, 2023. I cannot believe I am 3 years and counting in. Each year seems to go faster than the previous. And I have to say I'm as buzzed and energized and loving the job, thank God, as much today as I was 3 years ago. And that's in large part because of just the joy that, you know, I have in being part of this extraordinary institution that you are part of, and a very strong sense of our purpose and what we do for the world and for the betterment of society. And you all play that part. And of course, I have such pleasure in meeting the students and the faculty, and then meeting our alumni, which you will soon become, as well as we go forward. So those are just a few snapshots of me there being admitted. Obviously, our former Chancellor, Chris Patton, traipsing back to my beloved Merton College for the lunch afterwards. But when it all began was back in 1985, which I am mindful is decades before any of you were born, apart from some of you I can see in the audience. And I came up as a fresher from a local school. I was born and bred in Kidlington. I went to a local comprehensive school there, Gosford Hill, which I'm still very much in touch with. And I was very fortunate to get a place as an undergraduate and then indeed graduate at Oxford at Merton College. And you can see me there right at the top. I was a rock climber, so I was pretty good at climbing. So only those who were sort of in the rock climbing mountaineering club were told to climb up the trestles and be at the top. So that's why I ended up there. Keen rower, keen sportswoman. This is my husband, who you should definitely get to come and do a talk, Miles Allen. He's a professor here in the university in the space of climate physics and very well known in that area. This is us young, not grey, not wrinkled, pre-children, pre all the stresses and strains of being an academic and running things. But this is us in the gardens of Merton in 1994. So we're 31 years married now and counting. So I did my undergraduate and graduate work here in Oxford. I then got really inspired during an undergraduate lecture. So the first thing I want to stress is the importance of serendipity, great mentors, and the willingness to take a punt. There's quite a lot of themes in that one slide. This person here is Professor Sir George Rada. He sadly passed a couple of years ago. He's been an incredibly important mentor for me throughout my whole life. He gave an inspirational lecture as an undergraduate to me, and I think this is a great example of why we have in research teaching intensive universities our frontline researchers still delivering lectures. I still give lectures to the medics 3 a year. And it's really important because that's when we bring the excitement of the cutting edge research to you as students, particularly to the undergraduates. And it also helps them understand that nothing is textbook, nothing is black and white, everything is changing, and they can be a part of that. And so it was through a lecture that he gave that I became just literally light bulb moment inspired to want to move into this new area of magnetic resonance spectroscopy as it was then. Oxford's got a great long tradition of being brilliant at MR. We were the physics department that developed superconducting magnets. Oxford Magnet Technologies being the spin-out company for that, and they're the main magnet companies for most of the companies that supply magnets around the hospitals in the world, Siemens, Brooker, Philips, GE Healthcare. So great origin of Oxford in our magnetic resonance. And what George was doing and his team were developing new ways of using that to look at the living biology in a living, breathing animal, including human animals. And that just was extraordinary. Up until then, biochemistry was all about medicine. You take some information from a test tube or petri dish and you try and extrapolate what might be happening in the actual human condition. That's a big leap from a test tube into the body. So to be able to actually just put the body, the person, the animal, inside a scanner and get information straight out and not have any of that interpolation was just phenomenal. So this technique was really exciting. Parallel, there was a team in Boston at Harvard who were applying some of the discoveries that actually were made in biochemistry around MR in the 70s, which was how to, without going into it, exploit these techniques to go beyond spectroscopy, to do imaging, and then to do this thing called functional imaging, where you could actually see the brain in action and you could start to map it. You could start to determine which bit of the brain was controlling how you do logic, how you see, how you smell, how you taste, how you feel, all the basic perceptions. And so this team in Boston was putting all together this basic discovery science that actually was happening in Oxford, just literally around the corner in biochem, and then did it first in vivo. And so I was just lured into going out there for a postdoc, took a punt, and had just an extraordinary time. There's a whole backstory there, but we won't get into that because of time, in terms of different job offers and decisions about what to do. But I picked this lab because it was the best place to go and be trained in that important postdoc period of my career, where I felt I would be stretched and pivoting using my skills in MR into a slightly different area, firmly moving me from what I'd been working on, which is muscle disorders and muscle disease, into neuroscience. And I could see that neuroscience in the 80s was going to be this new phenomenon that was going to take off. And I was becoming expert in a technique that was going to be pivotal in actually opening up the black box to the human brain. So for a set of different reasons, again, decision-making, keeping doors open, looking to see where you're going to be stretched and pushed, I took a punt and went to Boston and had just an extraordinary couple of years. Married Miles halfway through, he came and joined me and was at MIT. So we just had a very magical time there. I've been very blessed coming back to Oxford. I've had throughout then my career subsequently affiliations with different departments as I've had different academic positions. So of course started at Merton, went to Harvard. I then came back, I was a junior research fellow at Corpus Christi. I then had my first tenured position at Christchurch in the Department of Physiology, Anatomy and Genetics. And then my chair, in the field chair, was held at Pembroke College. So I feel very blessed that I've had this a broad range of experiences of different colleges. I'm very pleased and proud that I'm an honorary fellow at all of those. So I have lots of affiliations and homes that I feel I can go to. But like with many of you, I'm sure here, if this is your first experience of an Oxford College, even if you might spend more time in another college, and I'm very public about this with these other colleges, which I love dearly, there's something very special about the place where you sort of spent your first formative period of time. So there is something very special for me about Merton, and that's relevant momentarily. All right, the human brain, it's phenomenal. It weighs 1.4 kilograms. It runs on a Mars bar or a Snickers bar compared to AI, which needs all these energy things. It's got 100 billion neurons. Those are the computing elements that do the job. It's got these other things called glial cells. That's Latin for glue. We thought they did nothing and just sort of provided protection for the neurons with the business end. We got that wrong. Turns out glial cells are really important and do an awful lot more in terms of controlling of both the nourishment, but also the fundamental blood delivery control to the neurons to make them work, and more than that. Those neurons obviously have to talk to each other, so there is about anything up to 1,000 to 10,000 synapses per neuron. You've got extraordinary collections, and during these really critical windows of development, so obviously during early pregnancy as you're developing the brain, early postpartum, and then a bit later on as you start to decline, you've got this explosion of growth and wiring of the neurons, and then the contraction. Just to put it into perspective, it's about the same as the number of stars in the Milky Way, and that's all in your head, protected by your skull. So it is a phenomenal thing that, of course, we've spent many millennia evolving to do what it needs to do. And what you can just see here are three little brains that are 3D etched brains that show you just how your brain has developed from birth. So that's a brain straight from birth, through to what your brain would look like in your teenage years, through to what it would look like in adulthood. Before then, potentially, as you get my age, it starts to go down a little bit. And so you can see it both grows in volume, but it also grows in its folding. And also within that, which you won't see, it also changes its wiring, its connectivity, the different ways that the different functions that your brain is going to perform are going to be connected. And you can see on this picture here, a beautiful, colourful picture. That's all the wiring that we can now detect, and that technique was developed again in the lab that I used to run. So it's a phenomenal organ, and one of the things that's really important in the broad field of neuroscience, not just the area of pain that I work in, is just how much we're learning through huge data sets like UK Biobank that's gathering information from 100,000 people's brains and then relating its structure and its function, its chemistry and its performance to socioeconomic background, whether you do physical activity, your nutritional background, your genetics, your different sort of cultural upbringings. And when you've got that number of brains, you can make statistical inferences about how your brain is developing and wiring and potentially becoming a vulnerable brain or a resilient brain that will equip you or not equip you for the rest of your life. We are learning powerfully and very politically sensitive things about those relationships, which are really very important. And you'll be proud to know that Oxford is at the epicenter of all this work. And this is going to profoundly change decisions that will be around the importance of early intervention, education, nutrition, socioeconomic status, because fundamentally, your brain's going on a pretty impressive journey from birth to 20. That's a critical window to shape it and sculpt it and wire it to make sure it's as ready for what then is going to be potentially the onslaughts of adult life. And I think we're only just scratching at the surface of just how important that journey is and what things we need to be doing to intervene. Now, good news is the brain's more plastic than we thought. It can change, it can still adapt in ways that we textbook didn't realize until we had techniques. So we love physicists, we love mathematicians, we love engineers, because they give us all these new techniques and tools that allow us to completely change textbook understanding of brain. It really was assumed once you got to that big brain, you basically just declined. And now we know that, take up a new instrument, do Sudoku, learn a new language, do anything, frankly, and keep doing it until you're in your 80s, because your brain has actually a phenomenal capacity to still rewire itself, to make itself, more resilient, and to learn new tricks. And we didn't know that because we just didn't have techniques, in vivo techniques, that could let us measure that, as people would say, learning how to juggle, for example, which was one of the first experiments that the team did. So it's a phenomenal organ. It's very perfused. Sherrington, who is around the corner, Nobel Prize for the reflex. You stand on a drawing pin and you withdraw. That's what he got the Nobel Prize for. Everybody claims Sherrington, Cambridge, us, London. He moved around. But of course, his pioneering work was done here. But he was amazing, and he did lots of different things, and things related to pain as well. He came up with the first idea of these specialized receptors that could detect things in the environment that are going to hurt you. But he also did a lot of work just to tell us the fundamental, what we call coupling. When the neurons work, that's computationally active. They need energy. So you've got to deliver oxygen and glucose to feed them. And obviously blood's going to do that. And I think people are always struck when they just realize how perfused your human brain is. So if you take somebody who's died and you perfuse through the carotids and the arteries some cement, so it gets into all the arteries, the veins, the capillaries, and then you rot away all the neurons and the glial cells, that's what you're left with. It's extraordinary, isn't it? So now you can see just how highly, highly perfused your brain is and why it consumes the highest percentage of your overall energy cost a day, because it's feeding all those neurons. You can also see the vulnerability that then is in the brain in the context of strokes and changes in terms of vasculature that might affect, again, outcomes there. But extraordinary, again, legacy here in Oxford, highly perfused. And this really is the basis of functional imaging. What Roy and Sherrington did, I always love the fact they went to the labs with three-piece suits back in the day and did experiments on themselves, because there wasn't really ethics committees then. But they worked out with no kit. I mean, I've got all these fancy magnets, right, costing tens of millions. They just had basic equipment like a strain gauge, cut out a hole in the skull and measured it, and worked out that when you do something, your basic perceptions are you see, you smell, you taste, you hear, and you feel, right? And that's how you interact with the environment. And those signals come in, you decide what that means in the context that you are in tonight. You make a decision how you feel about that, and you act back out. And that's sort of your little decision-making compute, basically, that's often making decisions based on what those sensory experiences are, things that you like, and avoid things you don't like. And you can reduce sort of the richness of humanity down to sort of that. And the brain and the head is bringing in all those basic sensory perceptions, and they all have specialized bits of the brain where you largely control the ability to see. So vision's at the back, sensing is around this strip here, memory is sort of a bit deeper in, etc., etc. So it's localized. And what was assumed is that when you did a particular thing like seeing something, there would just be a general increase of blood to the brain. But what Sherrington proved in the 1800s was that actually there was local delivery of blood just to that bit of the brain that needed it. You then have to go pretty much 110 years later before then functional imaging and all these different imaging techniques came along, and what we're doing in order to put it all together in these clever experiments in biochem that then allowed you to use what we call a functional mapping techniques, you can see it on the bottom right, to map where there is local delivery of oxygenated blood, which when it's oxygenated, for those who are really interested, the hemoglobin has then a diamagnetic ion atom as opposed to a paramagnetic ion atom, and it just distorts the magnetic field that you're lying in this big magnet. So what we're able to do is, because of Sherrington's observation that there is local delivery of blood to that bit of the brain, and there is this phenomenon that distorts the magnetic field that you're in, which we're taking images in, we can map that distortion to that region, and that's how we map. And it's down to like the millimeter resolution. So we have all these amazing techniques available to us that we can look at the human brain in vivo, take you up to the lab, and we can look at your chemistry, we can look at your wiring, we can look at your grey matter volume, we can get you to do tasks. I can burn you, electrocute you, stab you, and I can see how you react to that and where activates in the brain that's responsible for pain. And that's what basically we've been doing for many, many years. In Oxford, of course, we're brilliant at those techniques, and we have some of the world's leading labs doing them, and we've developed and built them. But we also have amazing other techniques that allow you to look at the brain at the timescale in which those neurons are talking to each other, so in the millisecond timescale. And again, these are a range of different techniques that we have. So we're in a really good place from a position where we really didn't know much in the 80s about the human brain, because we had no techniques to look at it, to a place 45 years later where we've just got ever increasing ways of assessing the anatomy, the physiology, the network activity, the neurochemistry of the human brain. And we can put it all together. And then with big data cohorts like with UK Biobank, we can start to make really interesting inferences about the societal, educational, all these different impacts on your brains as they develop, and then how we can make them resilient going forward. I spent my career both in parallel helping develop some of those techniques and apply them and push them and want them, as well as then mostly applying it into the study of pain. Why pain? It's one of the biggest medical health problems we have in the developed world. It is something that you all experience. You've all, I'm sure, experienced acute pain. You've all touched a hot pan on a stove or cut yourself with a paper cut or had a sports injury or whatever it might be. 1 in 5 of you, very likely, maybe not this demographic, but a bit older, will have a chronic pain condition. So that could be migraine, it could be back-related pain, it could be joint-related pain, it might be something more serious related to other conditions. So it's a basic phenomenon of an animal to experience pain, because it's a really important warning system that tells you something in the environment is hurting you or potentially going to hurt you really enough to kill you. You better make a decision to do something about it. So it goes back to that decision-making thing that we are, which is to seek things that are nice and avoid things that are not nice. And pain is a fabulous teaching signal in that context. And that's why a lot of computational neuroscientists use it as a tool for learning related phenomenon, because it's a great teacher. And that's why we have it. It means that you've learnt, you don't remember when you learnt not to put your finger in a candle flame, but none of you would do it, right? But you learnt really early. Either you were told by your parents or somebody not to do it, or you experienced it and didn't do it again. So these things are really important. You learn how to avoid them. Chronic pain is just a system gone wrong. It is just a disease, it's a problem, and we're reclassifying it, and I'm not going to get into the sort of details, but it's a huge problem. 1 in 5 of the adult population have it. They have then comorbid depression, anxiety, sleeplessness, so it often results in job loss. It often results in family breakdown. It is devastating. It's one of the biggest, costliest problems for society. It's about 200 billion euros per annum in Europe for the treatment and management of chronic pain. And if you don't know anything about health economics, that's enormous. And it's multiple hundreds in the US. You can put it roughly at 1.5 to 4% of GDP is spent on the treatment and management of people in chronic pain. And frankly, we don't really have any good drugs to treat it. So most of the drugs we have been discovered for other things, and largely what they're doing is just dampening down neural activity. They're not targeting the mechanisms that have gone wrong in chronic pain because we've had the wrong paradigm. So they work probably in about 30% of patients, but at best they're just dulling it. So this means there's a huge opportunity, and that's why it's one of the big areas of research. It's one of the big areas of focus. It's also why there was an opioid epidemic in the USA, which you, I'm sure, have heard about. You've heard about the empire of pain. You've heard about all the distortions and the skullduggery and the desperate things that went on in the context of Purdue Pharma and etc. And this, I mean, I could give a whole hour lecture on the opioid crisis in the US, but I'm just going to say what it has done is, of course, highlighted that there is a lot of chronic pain about. And in the desperation of patients not having their chronic pain managed, the desperations of physicians frustrated they can't manage them, overprescribing of opioids that aren't going to work long-term in chronic pain, brilliant for acute pain, absolutely fantastic, never found anything better in several thousand years, but not for chronic pain. In fact, you'll make it probably worse. And of course, they're addictive. So you get what we call tolerance, habituation. You get all the sequelae and the side effects. The reality for the opioid epidemic is that actually the number of prescriptions for opioids, because of the awareness and the campaigns in the US, was going down from 2011, right? So everybody got on top of it, started to minimize the access, but the deaths went up. Why? Because once you've created a culture societally of lots of opioid use, and there's a lot of them prescribing and giving people 500 OxyContin when they leave hospital instead of in the NHS, God bless us, you get a couple of paracetamol. But you go back, you've got this great big tub, right? You're going to maybe, you know, and it's the McDonald's factor. If you've given a bigger burger and a bigger bag of chips, you'll eat them, right? So if you give them more OxyContin, you're going to eat more of them. But there'll be some left. Medicine cabinets packed, kids take them out, students, pill parties, combine it with booze, you stop breathing, you die. So you look at Prince, you look at Whitney Houston, you look at all these deaths, and it comes back to, again, this lethal cocktail and this over-prescribing, this over-availability, this then social appetite that got created culturally in the young, and then the street chemists come and they make fentanyl. And there's a vial of heroin. If you take that vial of heroin on the left, that will kill you. To the right of it is a few grains of fentanyl, and that will kill you. And that's laced in cannabis, it's laced in all sorts of other things. So don't take drugs, because you don't know what you're having, and that will kill you. And that's why 75 young people your age, every single day in America, are still dying. Because it's not the prescribing now, the opioids, it's the fact that the street chemists have come in and they've made these incredibly potent opioid, that's just off the charts, and it's killing the young. So, you know, tale of woe there, but the point is also from the pain point of view, it put a spotlight on pain's a big problem, and there's a lot of people in pain, and we're really bad at treating it, so let's come up with some decent drugs. So that's what we've been trying to do. So the basic building blocks in neuroscience of every perception you have, you see, you smell, you taste, you hear, they are all built on what we call a three-order neuron system. The first neuron takes the information in from the environment, hearing, auditory waves, visual waves, pheromones for your nose, things we're going to deliciously eat tonight, right? It gets passed on, that's called transduction. It gets passed on then to what we call transmission, get it into the central nervous system, and the final thing is perception. The brain gets a hold of those signals, puts it together, and you say, oh, I saw something, I smelt something, I heard something, I tasted something, I felt something. That's your basic building blocks. And we've learned a lot. We had, in fact, Arden Pataputin here just before Christmas. He got the Nobel Prize in Medicine for discovering all the receptors in your skin that can talk to things in the environment that can hurt you, which basically fall into three categories if you think about it. Things that hurt you in the environment that make you learn that pain to avoid it fall into the thermal domain, you get burnt and you get frozen. They fall under the mechanical domain, cut, hammer, that type of thing. And then it's the chemical domain, so bee sting or, say, chili peppers. Many of you, I'm sure, have chopped chili peppers and then rubbed it in your eye and really regretted that, it really hurts. Yep, these are chemicals. And often, I won't get into it, but this is what I teach for the medics still. It's really interesting how we've learned a lot from, sadly, people who can't transmit. So there's people who have what we call congenital insensitivity to pain. They don't feel any pain. Back in 100 years ago, they earned money by doing circus tricks. They would jump off buildings this high, they'd crash, well, they wouldn't get out because they'd smash their legs, but they certainly didn't feel any pain. And of course, these were freaks. They died because they didn't have any acute pain. They didn't realize that they had internal bleeding, sepsis, and they're gone. Obviously now they're more looked after by our wonderful clinicians. And you can see here an X-ray of indeed somebody who's got completely smashed legs, felt no pain. So what we've learned about these genetically is they don't send the signals into the central nervous system. They transduce, and they've got the machinery to perceive, but if the brain doesn't get it, you're not going to feel pain. And that's what and so that's now a new molecular target for drug discovery. Same for the transduction. I've been obviously working at perception. So the challenge of pain is it's a really slippery thing. You think it's nice and straightforward, and Descartes, wonderful man as he was, thought it was very simple. What happened in terms of the injury from the environment, the burn, just sent up the spinal cord, faithfully to the brain, and it's a one-to-one relationship. So how much you get injured is how much pain you feel. And what we've learned over the years is that's not true. It's a highly nonlinear relationship. You can have huge injury and no pain. You can have no injury and huge pain, and everything in between, because it's a highly nonlinear relationship, because that three-order neuron system has had evolution put all these points along the journey where it can turn the signal up and down. So this is important because in courts of law, you can't make a judgment about whether somebody should get disability access for a pain syndrome based on looking at an X-ray. There's no way you can tell what their perception is going to be based on what you can physically see with crude techniques out the periphery, because it's just not a linear relationship. And I've spent my career in that gap, using non-invasive techniques to understand the non-linearity and to explain it. I'm just going to bring that to life with a few experiments. So just to drive it home, I can take all of you, I can burn you, right, with devices I have, with my phone. I don't do the science anymore because I'm obviously running a university, but I had a lovely torture chamber with all sorts of goodies in there. And I promise you, I can give you exactly the same injury every minute, and I can then distract you. I can make you attend to it. I can make you happy. I can make you sad. And I can completely change how you feel and report that pain is, from really excruciating to not so painful. And I can do it really easily. I can really manipulate that malleability. And what I'm doing is I'm changing the pattern of activity. And if I change the pattern of activity, sorry, this is not Pixar animation, it's as good as I can do. You can see the pattern changing to the same injury input. And if I change the pattern of activity, I change your perception. And so the environment into which those signals are coming from the injured bit of the body, if it's a depressed brain, a happy brain, a distracted brain, an attentive brain, it will profoundly change how those signals are going to be processed, and it changes the perception you have. And I'll just show you some fun examples of that now. So first experiment we did is we took a bunch of you, we put you in the scanner. We almost simulated going to the dentist and being completely panic-stricken. I won't go into how we did it. It's a fun design. This is where the intellectual bit comes in these experiments. But we can really simulate that you're really panic stations when you know you're going to get this next stimulus. Frankly, I'm giving you exactly the same stimulus. I just make one condition where you think, oh, it's that one, and the other one you think it's going to be the bad one, but it's not. It's the medium one. But because you think it's terrible and you're fearful and anxious, as you would be going to the dentist, you rate that same medium pain as more painful. And then what we're doing with the imaging is finding out, where's the amplification going on? Because it's certainly not going on in the burn of the skin. I'm giving you the same injury, but I'm doing it in your head. And then we discover these little areas around here, which is where there's the anxiety amplifier, turns up the volume. This is, anybody know about spinal tap? Going to 11, it's just like turning up the volume to number 11, right? And I can do it right inside your head, just by changing the way you think you're getting a signal. Context matters, expectation matters. And it's not just changing the way you describe things, it's changing the actual neurochemistry and neurophysiology of how your brain is working, because of the beauty of these circuits you've got inside. It's phenomenal. This one in the middle here was a fun experiment. That was where we took a bunch of students, often we use students, we put them in the lab, we burn them, and they're listening to some lovely music. So they're in what we call a neutral mood. They're listening to some lovely Dvorak, they're reading statements that says, I sit on a chair, cherry is a fruit. Nice neutral things. It's a classic sort of what we call mood induction. And then we make the mood go a bit dark. So we start playing Prokofiev's Russia Under the Mongolian Yoke at half speed. And if you listen to that at half speed, it is miserable. And so we make you feel really sad. And then you read statements inside the scanner, my life is a failure, I have no friends, I am worthless. And we statistically make people sad. And then we then burn you again. So what you can see on the bottom row is your brain is a lot more red and yellow. If your brain is a lot more red and yellow, it's a lot more active, right? And I've not changed the burn. I've just made you sad. And look how much more brain activity I get. It's almost like giving you another 5 degrees centigrade, but I don't have to do it in the periphery. I can just change the mood of your brain. That profoundly changes the way you process those signals. It changes the whole perception in both an intensity dimension and a sort of affective dimension. Why was this important to do in those early days? Because it was proving this nonlinearity. It was also proving something, I don't want to get all glamorous, but a bit more profound, which is that these psychological features of mood, cognition, context, expectation, they didn't just change the way people orally described their experience, they changed the fundamental physiology of what was going on inside your brains. And that was not known. And so that then provided more opportunity to work with patients, with psychologists, because pictures are really easy to understand. And you could just say, if you don't keep things that are positive in your life and doing things that bring joy, look what your brain's going to do. That's a lot more pain. You don't need to do that. You've got power to try and lift it out. So keep things that are positive in your lives, etc. So again, these can have repercussions. I'll skip over this for time. It's hard with pain because of that complexity. And again, we have very crude ways of measuring it. We either get you to say on a simple scale, 1 to 10, how much pain are you in? Well, how simple is that? When you've been in pain, your life is over, your partner's gone, your whole identity is chronic pain, and then we're distilling all that suffering into, on a 0 to 10 scale, where would you like to put your number? Doesn't quite capture it, does it? Some kids can't talk, so people can't talk to children, so you give them a smiley face or an unhappy face. If not, you observe and infer from behaviour. We've all got our own pain stories, but we're biased on what we think. And we're generally less empathic than we think we are. We all think somebody's probably exaggerating and being a little bit of an exaggerator when it comes to their pain, because you can't help but bring your own bias into the equation, and your societal bias, your cultural bias, your own pain journey. It's really important. This is what I drum into the medics when I'm teaching them. You've got to be objective, not bring any of that into the equation. We've got a whole martyrdom. I'm a practicing Roman Catholic. My goodness, I can say it because I'm a practicing Roman Catholic. Wow, we were masters at the whole, suffering and martyrdom and thing. Actually quite genius. The more you suffer in this world, the more you'll have reward in the rest. Whilst there were no drugs to treat it, actually it was a good trick. It worked really well. Sort of got the whole placebo thing going. But we've just got these traditions that you don't realize you are being bombarded with that bias you about your attitudes. And then when people are comatose, vegetative states, demented, they can't answer. They're not moving. So you have to use indirect physiology to say, are they in pain? And it's really difficult. So here's Serena Williams. Hands up if you think she's in pain. She had a shoulder injury at the time. Oh, you're too smart. All right, hands up if you think she's going, yes, I've just won that 3rd set. Yeah, that was actually it. Most people, when I do this, go the other way. You're too smart for me. The point being, it's ambiguous, yes, and it's difficult to infer, and you can be really wrong about it. And so that's why we need to develop pain biomarkers. You can't take a blood test. I wish I could. I wish I could take a blood test and put it in a sensor, and it would tell me this person has 8 out of 10, it's for this reason, and you give them this drug and it'll work. It's just never going to be as simple as that, as it is for some other conditions. So we're constantly trying to develop an understanding of the mechanisms, what's going on, and the biomarkers that we need to develop to make that happen. There's many different types of chronic pain, from nerve injury to musculoskeletal to what we call functional, because we don't quite know what they are, but they all have quite a lot of things in common. So despite coming from many different types of conditions, they share symptoms. Often it's like touching with clothes, burning sensations. It might be just spontaneous stabbing or electrical shocks. Sometimes it's sensations that people haven't even experienced in their life. They don't have words for it. But there's common symptoms despite different patients. And the important thing is that means that if you share symptoms despite starting from a very different type of condition, it's become something other than the condition. Whereas historically we were teaching the condition and not recognizing that there were mechanisms underlying those symptoms that were shared mechanisms, even if it was pain due to getting diabetes, or pain because you fell off your bike and ripped your nerve, or pain because you got cancer and you went on to chemotherapy and they affected your nerves. All these different classic pain conditions, profoundly different starting points, but they share mechanisms, which means once they've transmuted into chronic pain, they've become something other. And that's the new paradigm, that's the new framework we think about it, that's the new biology we're targeting, and there's lots of wonderful discoveries coming along. One of the big symptoms patients have is just this constant throbbing, ongoing pain. And again, we developed a new imaging technique that allowed us to identify where that was mostly represented neurally. We then worked with the neurosurgeons to go in and target that neurosurgically. We just actually published that paper as a completely novel target. So again, you know, a very quick and simple example of where you do a new technique, you apply it to understand a profoundly basic neuroscience question, but you can very quickly translate it, if you've got the right interdisciplinary environment, into a novel surgical target and solution. And we've done that sort of hands over. One of the areas that I spent a lot of time on, and this is really important for sort of understanding placebo and other things, is the descending pain modulatory system. So this is this incredible system with what we call a good cop and bad cop. What you want to have is lots of good cop and not so much bad cop. And what does it do? Well, it basically talks down to, from the brain down to the starting point of where those damaging signals from the injured body are coming into the spinal cord. And basically either it inhibits those signals so they don't come up to the brain so you don't feel pain, or it amplifies them. Now, we've done a lot of work on the amplification because that's underpinning for chronic pain, and that's a whole sort of translational story, and we've proven how important that is for chronic pain maintenance, and that's a target to go for. But the good cop is the one that is where you would be using this in your everyday situation, because this is the fundamental mechanism that's as old as time, uniquely there for pain as a sensation, not there for any of our perceptions. Exquisite machinery in the brain. The sole job is to go down and block those signals in the spinal cord so they don't come up so you don't feel pain. Why? Because back in the day, if a lion has bitten half your leg off, you've got two options. You can lie there writhing in agony, and that's going to kill you, or you can have the pain blocked in order to make the decision to get away. And so that's why you've built this system, and it's phenomenal. And it is the basis of why when you are distracted from pain, it doesn't hurt so much, listening to music, watching a movie, taking a child to have their vaccination and distracting them. It's the basis for the placebo effect. You're probably a Harry Potter generation, and you'll know the Harry Potter potion. Placebo is really good. It's in fact probably one of the better pain relieving mechanisms we've got in the world right now. We don't use it because of all the confusion over at the ethical side of it, but it's phenomenal. And basically it hijacks the system. There is nothing special about placebo, but it is real and it is physiological, and it uses this ancient old mechanism of this good cop, and it basically just blocks the signals coming in. Now, the problem with placebo is you've got to deceive somebody, you've got to mount this expectation that this thing is going to work, and then you give them the dummy pill, right? The saline injection or the sugar pill. You can't ethically do that in the clinic. But of course, what placebo manipulations are doing are managing people's expectations. They're manipulating people's expectations. They're telling them one thing and mounting up the physiological response, and then you do something else, but the expectation is still driving the physiological response. Well, that's what's happening in every single patient-physician consult. Is you go in, you tell the doctor everything that you've read probably on Wikipedia or Google. They then dismiss most of the nonsense you've come up with and set up new expectations. You walk out the room, you'll have a treatment, could be a drug, could be something else, and it's the combination of the drug, the treatment, and your expectations as they have been set that actually dictates the outcome in a real physiological way. So we wanted to take that as a real life example of proving that this placebo thing that's really important and really powerful, but you can't use it. But actually what you could do is learn from it and take the expectation manipulation and then bring it into the therapeutic setting. So we did this experiment. We took a bunch of students again, put them in the scanner. They just have to reliably tell me every time I burn them, how painful is it on a 0 to 10 scale. They don't know what I'm doing. They just know that at some point they're going to get a fast-acting opioid, right? Which, as I said, is really good acutely. They don't know when I start and stop the infusion. They don't know whether I'm turning the heat down or turning it up. They just need to reliably tell me about the rating. So in they go. 15 minutes in, we start the infusion of the opioid intravenously, but we don't tell them. We keep taking the pain ratings to the burns, imaging the brains. We then go in with the big coat on, the white coat. We're going to start the infusion now, check your blood pressure, check everything's fine. They mount positive expectations, thinking, great, I'm going to get the drug now. They've been on it for 15 minutes. They just didn't know it. Positive expectation. We take their ratings, we're imaging the brain. We then go in again, the big white coat, right? We're going to stop the infusion now, so sorry, the pain's going to come back, and they go, oh, it's going to be awful. But we don't stop the infusion, we keep it going, right? So what happens to the rating? So basically, same pain, right? Same injury, same tonic dose of drug. All we're doing is changing expectation for what we call hidden, open, and then hidden again, right? And this is the ratings. So the first bar is out of 100, it hurts. Trust me, that's good, strong pain. It drops. You get sort of a 10-point drop. That's the true pharmacodynamic efficacy of that dose of that drug. You then tell them they're on the drug, and you get double the points of analgesia for free. That's your placebo mechanism, and it's for free. It's better than the drug. We actually thought when we said we'd stop the infusion and we didn't, and we mounted what we call nocebo or negative expectation, we thought we'd reverse just that open to hidden, but it went right back to baseline. So the anxiety, the negative expectation was enough to override the actual drug that you're giving them. And that is the power of the mind and your brains over really hardcore physiological mechanisms and things. And that's a really important illustration, but very important in the context of, again, how we manage patients. And then you can image and show what's driving it. And we showed that those papers we published ages ago, the little anxiety amplifier was the one that was driving them back to baseline. And then it was the frontal cortex driving the good cop, was driving the positive expectation. This led my surgical colleagues, who I'd done a lot of hips and knee surgery studies with in the context of the bad cop. And this is the bravest experiment ever. Wonderful colleagues up at the Nuffield Orthopedic Hospital, whose whole clinical career has been on this shoulder pain syndrome, where there's this little bone in this acromion joint and they shave it off, and that relieves, in theory, the pain. The issue with surgery is it doesn't often need the same control arms that you have to develop a drug. So if the surgeons can just make a sort of pitch as to why they want to do something, and they can just go and do it. Now, I'm fine with not needing a placebo or control arm. If you're going to take a tumour out or fix a bleeding vessel, go ahead, get it done. But if your surgery is targeting a subjective outcome, and pain is a private subjective experience, you have got to build in a control arm. But ethically, you can't do pretend surgeries. But we did this study. It took 10 years. Arguments with the ethics committee, different physicians dropping out in different clinical centres, because this is one of the biggest private practices, and they were worried, I think, about the results and what it would do. But my colleagues, unbelievable, up there at the Nuffield, particularly Andy Carr, took it on. This was his whole career. And he worked with me on all the other work and said, you know what, I wonder whether my surgery, my whole life has been a placebo, or whether actually there's something mechanistically about this. So we did the experiment, and it turns out it was placebo. So 3 groups, one does nothing, that's the green. Now, I don't know why my surgeons do this. When it goes up, it means it's better. So they're getting better. They start in a lot of pain. If the curve goes up, it means they're better. The green is after 6 months and 12 months. No treatment, they're better. The blue and the red were the 2 groups that came into the hospital, had the full shebang. They had the anaesthetic, but only one of them actually had the operation and actually did it. And the other one just pretend. No difference at 6 months, no difference at 12 months. You can imagine this caused a riot. He was public enemy #1 in the surgical field, but he wrote it out, and now multiple groups have confirmed it, and now lots of placebo surgical trials for pain are being done around the world because of these results that show you better find out. Now, if you need to come in and do the whole, come to hospital, have an anaesthetic, because that's what's going to drive the placebo effect, that's fine. But let's be honest with the patients. What's actually driving the pain outcome? It's not the mechanism that they thought it was. Okay, last tiny bit and I'll wrap up, and then the nice fun pictures of just my life. So we've done a lot of anaesthesia as well, because obviously it's the most powerful manipulation of the brain. And we've done a lot of work to understand and informed from how we can look at the brain. We've done a lot of work, obviously, with all the data we've gathered over the years. We've done a lot of machine learning. So we've put loads of data into different, we've been doing machine learning for 20 years, where we just train up algorithms to read brain patterns. Are you in pain? Are you not? So I have a very good sense, if I'm looking at a brain, and independently with machine learning, as to whether that person's in pain relief, in pain or not. You can take that then into a state where they can't tell you because they're fast asleep, right? They're anesthetized. And one of the big problems with anesthetics is we don't have a brain-based biomarker for perception loss. remarkably. So when you go into the operating room, largely the physicians, the anaesthetists, they're brilliant. They will be looking at your respiration change, your cardiac change. They'll be dosing you based on your height, weight, sex, and age. And all of that's done on population. You're actually putting the brain asleep, but they're not looking at the brain. And I'm telling you, I've spent 35 years looking at people's brains. It has nothing to do with your height, weight, sex, and age. So it's all sort of wrong. And generally it works because everybody's given just so much anaesthetic to make sure they don't move and they're fine, which means we're probably titrating too much, and that's a problem too. So we stumbled across a discovery, one of the hardest sets of experiments we've done. It's combining EEG with functional imaging, really technically difficult. You're putting people asleep in the scan. It's very, very hard to do. This is the only little bit of science I'm keeping going because we're commercializing this discovery. But we just came across something really profound, which everybody did, but it was unique to their brain. And it's these slow waves that you mount when you go to sleep. Turns out anesthetics produce them, and everybody produces a certain amount that basically is proportional to how much grey matter you've got, which just goes with your age. So it's this beautiful, simple biology of a relationship between how many slow waves you can make. And even if you give somebody more dose of anaesthetic, they will only make a certain amount of slow waves, which you can see built up on the red, but the dose of drug in the black is going higher. But that's it for subject 1. Subject 5's less. Subject 6 less. Everybody did it. Everybody, the moment they fell asleep, as the dose of drug went up, which is the black line, they all stopped responding, they fell asleep, and then they mounted these slow waves and they hit their peak, even though the drug went up. So then we combined it with the functional imaging to work out, well, what does that mean? And what we found was this is where you have perception loss. So it's brain-based, it's personalized, it's beautifully simple in its biology, it's the final gateway of where the brain shuts out perception, and it stops external things coming into the brain being processed. So that's the point you want to titrate to and hold somebody at, because that's your exquisite point of personalized anesthesia. And so we've done all these experiments, clinical trials. I've even got my husband Miles involved doing a real-time measurement. I've even got my son John involved at one point to get this over the line. We're still pushing this. I'm skipping over these because you just need to know we finished 2 big clinical trials. We're hoping to commercialize this. It works brilliantly. And to this day, it's the only brain-based individualized. So it's great for the translational point of view because there's hundreds of millions of anesthetics given every day. And my goodness, we need to start doing it brain-based and personalized. But there's this really interesting basic neuroscience question because, you know, I don't know why more people are not working in anesthesia neuroscience, because the most profound outstanding question in the whole of neuroscience or science is consciousness. And what does it mean? And can robots have them, et cetera, et cetera. And, you know, there is so many great tools now that we can start to explore that with. And here we've got a point that you can take the brain to where really profound things are happening that you can start to play around at in terms of understanding how does the brain fundamentally go into an unconscious state and stop perceiving the world and then wake up and do that. I'm mindful probably dinner's needed, so I'm going to just whiz over these final few. The backstory. Okay, that's the science. I'm giving you just a flavour of just some of the experiments that I thought might be just more interest and more generalizable as opposed to the nitty-gritty physiology. But to do all that, you've got to actually build labs. And this goes back to, again, some really key mentors that I've been very fortunate to have: former head of neurology, former head of experimental psychology, former head of biochemistry, who was my supervisor. Back in the day, nobody had computers, only Alan could type, most wrote, and you just wrote to the MRC and said, you know, we'd like to set up this imaging centre, would you give us £1 million? And they said, sure. They sent a cheque back. I wish it was like that now with UKRI. I wish grants were so easy, but it was the good old days. So we set up this imaging centre. I came back. This is me with George. This is just before he passed away. I went to see him a couple of years ago. And I came back to help build it. So here I am, geeky there in the yellow shirt, at groundbreaking with the trowel as we broke ground up at the hospital. We built the lab. 3 phases later, we extended it. Then we built another lab. I was part of obviously founding it, and then I directed it for 10 years. It's been probably the most important part of what I've done scientifically alongside my science in terms of facilitating science, just because of the hundreds of people that have come through that imaging centre and the physics we've done and the image analysis that we've done, and it's still one of the world's leading imaging centres. You have to raise tens of millions to get all the magnets in and make sure we've got state-of-the-art kit. For our physicists, I don't know why physicists love imaging fruit. I think it's because it's got water in and it's symmetrical, but when we got the new 7 Tesla, which costs £9 million, they kept showing me pictures of pineapples, and I'm like, well, I'm not sure that's what I'm going to tell the MRC we're doing with the magnets. But eventually we started to do some beautiful, beautiful pictures, and you know, a lot of effort goes in behind the scenes before you can even do the science. As Kristalina said, I then ran the department. So at that point, I was running the imaging centre. I was running the anaesthetic department because I had the chair there. And then we merged because we then moved the Radcliffe Infirmary up the hill, and we merged and we created the Nuffle Department of Clinical Neurosciences, which is one of the biggest clinical neurosciences departments in Europe. And we merged that, bringing all these things together. It's been a great success. Chris Kennard was the first chair. I then headed it for several years before I went back to where I began. And this is the picture of Kristalina, who's given me a lovely bunch of flowers when I left the department as its head, and Kevin Talbot brilliantly took over. And why did I stop running a department for running my old college? It was to give back. It was as simple as that. Merton profoundly changed my life. It gave me, and the education here in this university gave me my whole career and my whole joy of being a scientist, and I hope what we've been able to contribute. So when it was suggested that I could go back and be a part of looking after the college, of course I wanted to put my hat in the ring, and I became warden, and it coincided completely with COVID. So it was a very interesting time to be warden. And then I was being cajoled into taking on the VC-ship. This is me knocking on the door at Merton and being admitted as its warden. It's obviously one of the oldest colleges in Oxford. It's a lovely, lovely place. And it took a lot to persuade me. It took half a year to persuade me to be VC, because I knew that would be the end of my science life, which it has been, and you can't go back as an experimentalist. You de-skill, and I didn't want to be a phony scientist and pretend I was doing it on the side of doing this job. So that was a big ask, to basically know that that was, for the first time actually in my career, I was closing a door forever. And I'd never done that before. And so that was a big ask. It took me a long time to be persuaded to apply. I applied on the condition I'll just find out a bit more what this being a VC would be. And then 6 months through the process of the interviews, I then became sort of, okay, actually, I wouldn't mind it. And luckily, thank God, I do love it. So that's great. And it more than offsets the personal loss of not being a scientist and the joy of that. So that's sort of the building, very quickly, the building bit. Final few slides was fun. Of course, I've got 3 beautiful children. And you're juggling that on the side too. First child was born in '97. I was building my pain imaging group, you know, to the point of like 35 people. My beautiful other son, John, was born 2001. This is when I got tenure. So Miles and I were both, you know, postdocs. We had a house and a mortgage. I was pregnant with my second child, and neither of us had a contract for less than another year away. These are stressful times, and that's why I've put such an emphasis in my tenure as VC for pain conditions, early career researchers, trying to really try and create a bit of stability, because we have been there and we felt it. It was a long time ago. It's much harder now for you, but it was tough enough then. So juggling all that at the same time, it was very busy. I had very elderly parents. My mother was dying of Alzheimer's. We were caring for her at the same time, etc. How do you get through it? Well, you're young and you're energetic, and you just get up and keep going each day. I mean, and there are just these pinch points. You look back now and you think, I don't know how I did it, but you know, you do get through it, and you do now with reflection. And I say, and I share this very openly, you will have these extraordinary pinch points in your life and career, particularly at this stage where everything's at you. And you've got to rely on each other. You've got to get support with each other, use and ask for help, ask for mentorship. People will very rarely say no. They will help you, and you will get through these pinch points, and things will get a little bit easier. I had then a third child, and I was very fortunate. I had 3 of the actually rare women at that point who were running departments. In fact, they were the only women running departments at that stage. Kay Davis, Angela Vincent. Kay is still a mentor for me, so I still very much talk to Kay as an elder, and a few others. Angela Vincent still comes to the department, I'm sure. Lynn Brindley was the head of Pembroke. So I was very lucky in having extraordinary mentors, both men as well as women. And it was really important to see the different styles of how they conducted, you know, committee meetings, how they set the culture in terms of what they wanted to achieve inside the departments. And I do feel very blessed that I was fortunate to have that. And they were pivotal in being able to juggle everything that I was juggling. But I wouldn't have done any of the leadership stuff had I not had a husband and a partner who was willing to take on, frankly, the lion's share of the family. And I'm always very public and open about Miles being absolutely 100% there. And there's no way, I'd have just done the science, that's fine, but to take on the leadership one, which does take up time and spare hours in the day, I wouldn't have taken that on. And I do enjoy those roles, and I find them very rewarding. I find it very rewarding to create environments where people can thrive. I really like that. And I find that personally as rewarding as any paper as I published. And I wouldn't have been able to do that had Miles not really taken on that lion's share and basically took my whole maternity leave at a time when it wasn't possible to have paternity leave. And that's why I've been setting up paternity leave, sorting that out as well for men, so to make it easier, because he had it really tough. He was the lonely man in the playground. I kept being asked to go to the morning coffee mornings, not Miles, he was the one picking up the children. It was a bleak time. And he was rare in physics, but they were great, and we had great heads of department that helped us get through those pinch points. They supported us. They allowed us to work out what was right for us, and we asked for the support, and we got the support. So we're forever grateful to those people for that they've done. It goes fast. So just enjoy each every day, make every single day count. I know there's some people here who know John and Jim and Colette, so I've put those pictures in just to make you laugh. Of course, the love of my life now is my dog, Jeffrey Biscuit, who is available for dog walks. Some of you have probably come with me on that, so sign up, there's a long waiting list. But they, obviously it's such a joy, and it's that side of your life that you mustn't shortchange. You will have to make sacrifices if you're going to juggle these things. It's an absolute myth and lie that you can have it all, and I never, ever have told any of my team members, and particularly women, I've always been very clear about expectations. It's tough, and you're going to have to make some calls and some priorities, but you will get the support. Ask for the support, and you will get through these periods and have more time back. But you will have to make some sacrifices, and that's something you only have to decide yourself. I keep saying by running, so I've just finished my big 6 world majors, marathons. I'm not running marathons ever again. I'll do 10Ks and halves. This is my beautiful daughter. I did London. I raised money for Alzheimer's, the 10th anniversary of my mum passing with it. This is my daughter and I in New York, and just recently in Berlin, where I completed my 6th, and then I'm still running, but I'm not running marathons. It's a long, long way. But it keeps me sane. And then this is just a fun picture that brings together which one of my last doctoral student, he's a brilliant artist, he's an anaesthetist now over in Boston, but he brought together, as I left the pain group and said goodbye to my science life, this was a gift they created for me, which just sort of brings together all aspects of what's been my career and my journey to life. So I'm very, very touched by what they did there. If you like what you hear, there's lots of sort of media stuff I've done, but these are the most important ones, the team. This is the team I have had the pleasure not just to be a supervisor of and a colleague of, but to have been part of their journey as just wonderful friends and individuals, and just been part of their lives. And so the team members that I've had over the years, extraordinary people. You are extraordinary people, and I'm telling you, it is such a privilege to supervise people like you and to work alongside you. And it's more than just that. You become part of our lives forever, and we're so proud of what you do, and we really value that friendship and that collegiality that you bring, and the joy we have in seeing you, grow up and have families, if that's what you're going to do, and forge your careers. So lots of different people who I absolutely must shout out, because they're the ones that have done all that extraordinary work. And again, each and every one of those, there's a whole story behind each one. I know each and every one well. I treasure them. They're like family to me. They really are. And of course, none of this would happen if people didn't volunteer to come and lie on the scanner and get electrocuted and poked and prodded and burnt. And of course, all the different people that have funded us. I'm not going to reveal my questions because I'm mindful that we're 15 minutes behind time, so I'm really sorry about that. But very happy again to, in the Q&A bit after a bit of food, to either go back to any of that, or if you want to broaden it out to sort of things about the university, that is absolutely fine too, or anything on my personal life, very happy as well to take on any of those questions. But I hope that's been a little bit of something and a nice romp through my life. Thank you.